Hi, Dr. Wang here. Today we're going to work on Lab 9.1. This is a pre-lab assignment for FOA experiment. In question 1 to 4, you will make the necessary calculation to make the yeast dilutions for plating. An explanation of this is given in the flowchart instruction video. From this video, please go to the 2:20-minute mark. Here, the video explains how to make the necessary calculations, the amount of yeast to use for the next dilution, and the amount of water to use for the next dilution. So filling the question mark in this flowchart basically are the answers for the question in Lab 9.1, question 1 to 4. So now let's go to question number 5. For this question, we will need to use the concept map we have made for the FOA assay. So I have the map over here. This map basically summarizes the genotype of transformed yeast and the interactions between different genes within this organism. When we look at the questions in here, we have 6 questions, basically asking for the performance of 3 different yeast strains under 2 different environmental conditions or 2 different types of media. So I'm going to summarize the genotype first and then summarize the 2 types of media. Looking at the genotype, this is the first strain, yeast AGY75 transformed with plasmid PMSH2. The second strain is AGY75 transformed with the mutant PMSH2, and the mutant MSH2 in this question is assumed to be non-functional. So basically we're going to put a negative sign next to MSH2, which means that it cannot repair frameshift mutation. And then URA3 gene is going to be non-functional, and then the enzyme is not present, and then toxin is not produced, and then yeast is going to grow. Right, so this is basically how to use the genotype to predict the final phenotype or growth pattern. The third yeast strain is AGY75 transformed with the empty vector. So the empty vector is basically PRS413, and then this plasmid doesn't carry any MSH2 gene, basically means that this connection doesn't exist. And then the performance is actually still the same. There is no one to repair the frameshift mutation, and then the outcome of that is yeast will be able to grow. Now let's look at the 2 environmental conditions or the 2 types of media present in this question. Well, let's analyze these 2 media first. Both media are missing a couple of essential amino acids. So those are histidine, tryptophan, leucine, and threonine. The 3 yeast strain we use in this experiment actually has the ability to produce leucine and threonine. So we can basically ignore the absence of these 2 amino acids in the media. So let's look at this histidine and tryptophan. So the gene to produce tryptophan is right here, and the gene to produce histidine is right here. So basically all 3 yeast strains have the ability to produce histidine and tryptophan as well. So now we are left with FOA in one media, FOA in combination with the lack of uracil in the second media. So the growth performance of the 3 yeast strain in FOA media, we have discussed this already. So this basically depends on whether the frameshift mutation is going to be fixed or not. If the URA3 enzyme is not functional, we're going to get growth. If the URA3 enzyme is functional, then we are not going to get growth. Right? So the yeast is going to be killed. So if we also remove uracil from the media, this makes the situation more complex because uracil is an essential nutrient. So we also need to look at the question whether the yeast has the ability to produce uracil for itself. So when the uracil 3 enzyme is not present, then uracil won't be produced. This means the yeast cannot survive. Even though there is no toxin to kill the yeast, there is also no uracil to support the growth of the yeast. So no growth. So now let's look at the situation where the URA3 enzyme is functional. So what's going to happen? When the URA3 enzyme is functional, the yeast will be able to produce its own uracil. So this is good for the yeast. However, if you go down this branch, toxin is produced as well. So even though the yeast is not starved to death, it's being poisoned to death. So either way, this is going to lead to the death of the yeast. So you can also see that basically the standard FOA plate has to include uracil as its component, because if you remove uracil, then yeast just cannot survive.